So thank you everyone for joining today. Welcome to the free webinar on metagenomics, where we will be discussing microbiome and its role in precision medicine. In this online program, we will explore the amazing role of microbes that we can see in human well-being, health, and diseases. This webinar is for students and researchers and will cover the aspects of metagenomic sequencing, analysis of 16S rRNA, and other aspects of microbiology, research using bioinformatics. I want to now introduce you to the program speakers. We have Ilya Brodsky, our CEO and co-founder at Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. Dr. Harpreet Kaur is a certified omics logic trainer and expert bioinformatician who will guide the hands-on sessions and assist the participants with projects and analysis during our upcoming training programs. Her area of specialization is cancer genomics and machine learning. And Dr. Mohit Mazumdar is our omics logic project mentor and an expert in big data computational analysis. Dr. Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. I now want to invite Ilya to conduct the session and take over the session today. Ilya, over to you.
Thank you, Bibisa. And let me share my screen. Hello and welcome to this free webinar. I'm excited to see all of you participating today. And I hope that what we will share today will give you a greater insight into metagenomics itself, and also hope to introduce some of the materials and resources that we will be making available in this upcoming program. So as Bibsa mentioned, metagenomics is a new and exciting way to study microorganisms in their natural environment. And so this approach allows us to really dissect microbial community structures, but we have to also be mindful of the way that the data is generated and how it is used in the analysis. Now, these communities or microbiota allow us to understand how different microorganisms coexist, shape their environment, and develop a relationship with the host. We can study this microbiota to explore species richness and investigate the functional roles that these microbial communities play in many areas related to human health and disease. So in this webinar, what you will learn about is the upcoming Omics Logic Metagenomics program that will be starting on November 17th. This online program is designed to be equally rewarding to both students that are just starting to learn about metagenomics and researchers that are already involved in microbial research. And it is especially suited for project-based learning on a flexible schedule. So we will combine a number of different elements to really facilitate a flexible schedule that would make it available around the world and will make it possible for you to continue and apply these tools and methods to your own research. The program itself will cover main aspects of metagenomic sequencing, analysis of 16S ribosomal RNA, and other aspects of microbiology research using bioinformatics. At the end of the program, all of the participants can opt out for independent or group research projects. In other words, you can apply the understanding of theoretical concepts and the practical tools to data sets that we will help you find and curate. So we have prepared a number of examples, but you can expand on those and apply bioinformatics tools that are accessible to those that do not have any technical background, don't have any coding background, And for those that want to learn how to code, we will provide you with tools and guidance on how to use those tools using our Python and Bash scripting. At the end of the program, when you have prepared your project proposal or maybe conducted a full preliminary research proposal, we will connect you with an expert that will help you evaluate the project idea and potentially develop a competitive proposal that you can continue and work on after the program is over. So what we're hoping is that the majority of you joining today will enjoy hearing about metagenomics and where the field of microbiome is going, as well as utilize some of the resources that we plan to make available this month. So for those of you that are interested to learn more in the program itself, I'm just going to ask Beepsa if you could put the Metagenomics, omicslogic.com link in the chat box. And as we go through this program, you can explore some of the details of the program itself. Now, in all of our Omics Logic training programs, we combine online interactive sessions, kind of like what we are going to be doing today, self-guided study materials that I will briefly touch upon, and practical assignments that you can run analysis and understand what goes into the different types of sequencing analysis methods, processing pipelines, and visualization tools. Altogether, this creates an immersive experience that we are focused on developing to provide excellent training in bioinformatics. Now, what we have found is that many elements together become an effective tool for thousands of students that we have worked with over the years and that have completed the Omics Logic training programs to launch them into the field of bioinformatics. So even if you're coming from a biotechnology, biology, or life science programs, this is the way for you to start incorporating bioinformatics into your expertise, which is a highly demanded skill set in any area of life science research. In today's session, I'm going to focus on metagenomics, speaking mostly about the bioinformatics analysis of metagenomic sequencing data. These methods for processing, analysis, and visualization of metagenomics data include detailed tutorials on the use of such popular approaches like KIME2, Data2, and Phyloseq. So these are different types of processing and visualization tools that we will briefly touch on today, but we will speak in much greater detail in the program that will start on November 17th. But before we begin talking about bioinformatics, let's see how sequencing technology has helped develop this whole field of microbiome research and has provided an effective view into the study of microorganisms, and how these data analysis tools developed for metagenomic studies are used today to characterize microbiota and allow us to ask important questions about these microbial communities and how they develop and evolve in association with other phenotypic conditions that we can characterize. Since the discovery of the microscope, humans have been learning about microorganisms that surround us. And as the resolution of these microscopes improved, more and more representatives of the unseen world have been discovered. The scale of optical resolution can reveal details of up to 1 micrometer, which is a typical size of a bacteria. Today, microscopes are tremendously powerful, allowing us to see smaller and smaller elements that reveal how the world of microorganisms functions on every imaginable surface, and importantly, how microorganisms actively interact with each other. So in this image, you can see a super high resolution achieved with helium ion technology that shows the process of bacteriophages, which are in green right here. So these green dots are bacteriophages that are attacking E. coli bacteria. The blue are the E. coli bacteria. So you can see how this interaction is happening in real life just between two members of this community of microbiome community, microbiota, representing a virus or a phage and a bacteria. Now, the connection between disease and microorganisms has been established in the 1800s, and already towards the end of the 19th century, laboratory methods for growing microorganisms in a lab culture were developed, helping characterize them with staining. These studies provided important information about bacteria function in isolation. However, it was soon apparent that most microorganisms, in fact, live in complex communities. And it was soon discovered that many members of these communities are actually not possible to culture in isolation in a lab. So today, we know that the microorganisms, or the microbiota in collection, that make up the human microbiome are not just unicellular organisms living alongside each other, but instead form highly regulated, structurally and functionally organized communities attached to surfaces as biofilms, with interspecies collaborations, as well as antagonisms that contribute to its ecological stability. Due to the advances in metagenomic sequencing and other omics technologies, and enormous databases of sequencing data from such communities has led to the emergence of computational metagenomics, which can help study the remaining 99% of unculturable microorganisms in the context of their natural environment. So you can see here a brief timeline, how in the mid 1600s, first observations of microscopic organisms were published, and then the connection has been made between disease and microorganisms. And then from the 1800s until now, this practice of staining and microscopy has been used to study different isolated microorganisms. Eventually, in the 1980s, unculturable microorganisms were started to be studied using DNA sequencing. In 2008, an important milestone actually started in 2007, the Human Microbiome Project was launched and has provided, as we will see in the future slides, an expansive database of sequences of different microorganisms. And so starting from 2010 up until now, so the past decade and onward, we are seeing the important and critical role of bioinformatics or computational biology tools to study the communities in the context of this era of multi-omics datasets. Now, the stability of these communities is fascinating, as many of us probably know from trying to battle such common skin conditions as eczema, acne, and dry skin. These communities form distinct communities. The microorganisms in a microbiota are interconnected and determined to maintain their community. Bacteria within a biofilm can communicate with each other by producing, detecting, and responding to small diffusible signal molecules in a process called quorum sensing, which confirms benefit for host colonization, biofilm formation, defense against competitors, and adaptation to changes in the environment. Quorum sensing activities in biofilms are also involved in the virulence and pathogenic potential of bacteria and are therefore an important factor in understanding and controlling bacterial infections, as they enable microorganisms and biofilms to become more tolerant of host defenses and antimicrobial agents. So inside each community, there's a balance, in fact, of bacteria, fungi, and viruses. And together they control species abundance and the activity of that particular microbiota. The human microbiota is estimated to be 10 to the 13th to 10 to the 14th of microbial cells, so trillions of cells, with around 1-to-1 microbial cells to human cells ratio. Even though it was previously reported that there is a much greater number of microorganisms, about 1 to 10, these figures have been updated not so long ago by Sander et al. In 2016. These numbers are derived from the total bacterial cells in colon, which is 3.8 to the 10 to the 13th of bacteria, the organ that harbors the densest number of microbes. However, each one of these microorganisms carries an important functional property in genes that contain genomic information on metabolites and proteins that these microorganisms produce. These properties and the genomic information about microorganisms have been a growing interest to the scientific community, resulting in over 80,000 publications over the years. The majority of these studies have been focused on the intestinal microbiome, but more and more additional relationships between the composition of microbiota and different conditions has been emerging. Since the 1990s, microbiome studies were significantly advanced through such major initiatives as the Metagenomics of the human intestinal tract and the subsequent Human Microbiome Project. These are tremendous, huge investments and studies that have been made possible through the advances in metagenomic sequencing. So as a result, skin, lung, gut, and even brain microbiota are starting to be characterized in great detail. Each new project represents vast opportunities as well as challenges to determine which microorganisms play key roles in the development of certain environmental conditions needed for healthy function of human organs and which can be actually harmful to such communities. Over the past decade, more than 1.7 billion has been spent on human microbiome research. Major projects are underway in the United States, European Union, China, Canada, Ireland, South Korea, and Japan. This investment has confirmed the importance of microbiome to human health and development. It is now known, for instance, that newborns receive essential microorganisms from their mothers. Moreover, the sugars in breast milk that infants cannot digest nourish babies developing microbiomes, which in turn shape their immune systems. The biggest investment by far, around $1 billion, came from the United States. About 20% of this has gone to two phases of the Human Microbiome Project, you can see right here, which is creating the research resources needed for studying the human microbiome. One of the main goals of the Human Microbiome Project, launched in 2007, was to create a toolbox of reference datasets, computational techniques, analytical methods, and clinical protocols. This seems to have been a major success. About 75% of the 2012 to 2016 NIH grant recipients for microbiome research outside of the HMP is working on more than 100 different diseases. This is cited reliance on HMP data and tools in their funding applications. So it's becoming clear that microbes are needed to support human development and maturation and to activate and maintain stability in the immune system and metabolism. But there are still many things that we don't understand. For example, we don't understand how these fundamental biological phenomena involving human cells and microbes co-evolved. What's even more important, some ecological concepts are not yet commonly considered in the studies of the human microbiome. These include how communities of microbes operate as a whole, how keystone species can pave the way for others by altering local conditions, and the predator-prey relationships between different microbes. Developing a new conceptual framework and applying it to the human microbiome will require much more collaboration between investigators working across disparate fields, including evolution, ecology, microbiology, biomedicine, and computational biology or bioinformatics. It will also demand significant changes in how data and other resources are distributed between scientists. And importantly, it will require a good communication between those coming from the computational field, those coming from the health sciences field, and those coming from basic research. Because right now, these are very separate areas of microbiome research, and we see that they all interrelate, allowing many of these findings to come and be tested and become clinically important. So we know today that the microbiome function is important for many areas of human development, health, and disease. And it's because of this huge investment of capital and human research time that we now know that the endogenous human microbial communities contribute to critical metabolic, physiological, and immunological functions that include differentiation, maturation of the host mucosa and its immune system, food digestion and nutrition, energy generation, metabolic regulation and control of fat storage, processing and detoxification of environmental chemicals, maintenance of skin and mucosa barrier function, development and regulation of the immune system and fine-tuning of its reaction pattern, or the balance between pro-inflammatory and anti-inflammatory processes, and the prevention of invasion and growth of disease-promoting microorganisms, for example, colonization resistance. So an important underlying technology that connects many of these studies are being answered using next-generation sequencing, as well as other types of omics data, for example, mass spectroscopy, proteomics, and metabolomics. Indeed, it seems that the future, the next phase of prominent microbiome research lies in the ability to integrate multi-omics datasets that combine next-generation sequencing, mass spec, and structural data with phenotypic information about overall changes in the community and their environment. So to understand the challenges and the changing nature of such data, let's take a look at some of this data and how it could be integrated. So here we can see an approach to integration where you can see the immune system and health indicators being studied in the response to different types of treatments. So you can study the before and after and time course conditions to understand how samples represent different types of microbes and their changes over time. And you can study that using DNA sequencing, RNA sequencing, so the expression of specific genes, metaproteomics, or which proteins are actually being produced and how they interrelate to each other, and importantly, metabolites. So all of this has to be integrated using data integration approaches that look at the detailed structured data sets and then finding network associations, which you can see right here, between the different species and the products of their function. This is an important and emerging area of multi-omics integration that has to do with precision medicine, which is really becoming an important part of many areas of omics research. So I'm going to pause right now and take a look at some of the questions that I see in the chat and answer any of the questions that you might have about what we have done up until now. Okay, so if you have any questions, and Bipsa, if you could help me identify any questions that have already been posed, I'm ready to answer any questions that people might have right now.
Okay, yes, Celia. So we have one from Modibo. He's asking, I'm working about autism. I would like to know the specific microbiomes associated to autism.
Okay, so Modibo, thank you for the question. Indeed, autism and other neurodegenerative diseases have been linked to different indicators. I think what's important to understand is that many of such studies are in their early stages. And one of the things that you can derive from data sets that are already available are some associations. Now, association is not necessarily causality. And so that's one of the things where we will talk about in a few slides from now. We will be talking about how such causal associations could be studied using animal modeling. So we will talk about animal models of human microbiome composition and how those have been used to study different psychological neurodegenerative diseases.
Thank you, Ilya. We have another question from Shraddha. My area of research is breast cancer. How can I study microbiome in relation to it or the influence of it in breast cancer?
Okay, excellent question. So in fact, breast cancer and other cancers have been linked to tumor microenvironment. There are many publications on these topics, but also importantly, how the microbiome is affected by different types of chemotherapy and targeted therapy, because many of the functions of the immune response are linked to microbiome. Many of the cancer treatments are related to the immune response. So those are important areas of research. Now, we're not going to definitively answer these questions in this short presentation, but in the course of the upcoming program, what we will do is we will take these data sets and publications that present the evidence from sequencing data, and you will be able to analyze those data to find such associations. So I think if that is an interest, I will definitely point today to how to easily find such data sets and what are some of the common tools that are used to find such associations.
Thank you so much, Ilya. And if you have any specific projects that you are going to work on, I just want to take 1 minute. You can actually reach out to us at marketing@pine.bio for any such queries, and we will be able to help you out because it's not always possible to address all the questions in this one session. Ilya, for the next question, we have one from Nupur. Are these networks a part of network pharmacology?
Okay, excellent question. So this will be the last question for now, and I'll move on after this. But network pharmacology seeks to establish how specific active compounds can affect the overall systems biology, which of course works as a network or could be represented by a network of nodes that are interrelated by the edges in the network. Studying such networks and the changes in the networks is a field of its own that allows us to understand how the perturbations of the network can help reveal some of these prominent and strong associations. So what we will be talking about today are some general approaches to such types of data integration and analysis in bioinformatics. And it could definitely be applied to pharmacological studies as well, where you take essentially drugs, combinations of drugs, different dosages, apply them to different individuals, and then study how those are how the responses of those individuals are changing because of their, for example, microbiome or microbiome host pathogen relationships. How do those actually are affected? So we won't talk about this topic in detail right now, but we will definitely expand on such studies in the context of the program. Now, in the current presentation, I'm going to move on for right now. Please continue posting your questions on the chat. We will pause for another few minutes after a few more slides to address some of the changes, some of the questions that you have posted in the chat. So thank you for your questions. So let's talk a little bit about one type of such data, metagenomics. Obviously, metagenomics is a predominant technique used to study microbiome because it allows us to study the sequences. Now, metagenomics as a field can be defined as the genomic analysis of microbial DNA from environmental communities. Metagenomics tools enable the population analysis of unculturable or previously unknown microbes. This is very important because only 1 to 2% of bacteria can be actually cultured in the lab. The ability to identify microbes without a priori knowledge of what sample contains is opening new doors in disciplines like microbial ecology, virology, microbiology, environmental sciences, and many areas of biomedical research. Sequencing-based examination of the metagenome has become a powerful tool for generating novel hypotheses. Now, metagenomics, as opposed to other types of omics, really represents a different approach. For example, genomics, proteomics, transcriptomics all focus on whole of a single organisms. But metagenomics allow us to study a collection of organisms that live in communities and interact. So we're really, when we are approaching the question from a metagenomics perspective, we are looking at how the community composition, diversity is changing in response to different types of phenotypic data. Metagenomic sequencing itself can be applied to environmental culture-free sequencing to study populations, as well as for detailed extraction of genomic variation and the expression of genes from the microorganisms after culturing in the lab, And oftentimes, when we speak about metagenomics, we are speaking about those two technologies actually combined together. So here you can see on top an example of you taking a sample from the gut and then looking at culture-free sequencing reads, where we can look at OTUs, which I'll speak about in a second here, to understand taxonomic classification. Now, importantly, in some examples, you can take and culture some dominant species or representatives of this community to then map onto the reference genome and understand variation in those types of sequences. So for example, we can discover how antibiotic resistance, how changes in the specific function of the genes produced by the most abundant species in the microbiome could be linked to this taxonomic classification to understand how the whole community is affected by such changes. Now, the important technology that we're going to be talking about a lot is what's called 16S metagenomic sequencing, which is a technique that has been fairly well established because of many benefits for microbiome studies. It is widely used to study microbiome directly in its environment, and it focuses on the 16S ribosomal RNA that has to be amplified with PCR amplification. The 16S gene sequences contain what's called hypervariable regions, also known as HVRs, that provide specific signature sequences that are very useful for bacterial identification in the taxonomy of all bacterial species. The benefit of amplification is tied to the nature of sequencing read length, which can maximize the effective output from affordable sequencing technologies. Now, we mentioned hypervariable regions. So hypervariable regions allow us to understand how different communities look at the different types of microbial organisms that are present. Now, there's too much of a diversity to really study those individually. So instead, we can characterize them by organizing them into operational taxonomic units, or OTUs. So this is an operational definition that is used to classify groups of closely related individuals. OTU classification can be done using several different computational approaches: hierarchical clustering algorithms, Bayesian clustering, or phylogenetic analysis based on multiple sequence alignment. Once those OTUs are identified, as you can see in this example right here, and they are categorized by some common variation that is common to all the different species that are present and carry this OTU, then we can apply mapping onto databases. And there are several databases that have been regularly updated and used in such metagenomic research. One of these is the SILVA database, which contains taxonomic information for the domains of bacteria, archaea, and eukarya. It is based primarily on phylogenies for small subunit ribosomal RNAs, or the 16S for prokaryotes and 18S for eukarya. Taxonomic rank information for archaea and bacteria is obtained from Berkeley's taxonomic outlines and from the list of prokaryotic names with standing in nomenclature, LPSN. Whereas eukaryotic taxonomy is based on the consensus views of the International Society of Proteustologists. Taxonomic rank assigned in the SILVA database are manually curated, and for comparison, it was seen that SILVA is one of the more reliable, consistent, and up-to-date databases compared to some of the other ones, like the Green Genes, the Ribosomal Database Project, or RDP, and NCBI. So it's important to understand what this taxonomy allows us to see when we analyze data, applying mapping onto such databases. For example, here you can see the hierarchy of biological classification. Many of the OTUs lack annotation at the lower levels, like species, genus, and family. So a lot of times, the characterization of a community can be comprehensively done at the higher levels, like phylum and kingdom. Now, in the phylum level, we can start looking at how different microbial communities vary between different organs. And here we're talking about some healthy microbiota found on the human body. So you can see here some examples of phyla, like Firmicutes, Actinobacteria, Bacteroidetes, Proteobacteria, and Fusobacteria, and how they vary between some of the commonly studied organs in the human body. Now you can see, for example, the colon as compared to the skin. The colon contains the majority of proteobacteria as opposed to the skin, which contains the majority of actinobacteria. We can look at it from this level to understand what might be the environment that is caused by such composition, and we can study changes in this healthy composition across many individuals to determine any effect of common changes in health and pathology that is observed on these organs. Now, importantly, the bacteria are not the only components of this microbiome or the microbiota. Importantly, we have also the virome, which is actually one part of the microbiome and constitutes all of the organisms that inhabit us, including bacterial and archaeal organisms, as well as the microbiome and other members of the meiofauna, such as protists and metazoans. Studies of the virome are also in their infancy because it has only recently become possible to see the virome in large sequence datasets using advanced bioinformatic tools that can detect relationships between viruses despite their extreme nucleotide sequence diversity. Now, the size of the mammalian virome is not even known. As opposed to microbiome and bacteria, the virome composition is not really that well characterized. Although our own cells amount to similar quantities as the bacterial members of the human microbiomes, human feces alone contain 10 to the 8th viruses per gram. Additionally, the mammalian virome is continuously updated through rapid evolution of viruses and through exposure to viruses in the environment or in other mammals that we interact with. Now, importantly, we can also apply metagenomic sequences to understand presence and composition of viral bacterial composition in a sample. So here you can see some of the common sequencing techniques that produce a characterization of some of the common prevalent viruses that also play a major role in the composition of the microbiome, because the microbiome is a protective layer and controls the replication of some of these harmful pathogens, but also in the different diseases that they cause. Now, some of these viruses are very prevalent in cancers, and they also represent an important part of the interaction with the human microbiome. So what we will talk in the context of the program a little bit is how some of these viruses, like the Epstein-Barr virus, human papillomavirus, and hepatitis viruses, cause important cancers that are a huge burden on the human well-being and health. So let's talk about a few practical examples. In the program, we will talk about each one of these examples in greater detail. And so after I go through these examples, I think I will be able to address some of your questions in a little bit more detail based on what the information that we present here. A big part of the Human Microbiome Project is the American Gut Project. So you can see here a link. Let's actually take a look at this link quickly together. So I'm going to just post this in the chat. So we'll go to this American Gut Project. What happened? Okay, so it seems like their website is down, so let's instead go here. So you can see how American Gut is a very interesting project because it actually involves not just a comparison of healthy and sick people, but seeks to characterize microbiome variability based on multiple factors that are self-reported by citizen scientists. So essentially, any one of us can participate in this American Gut Project by paying for a small fee for sequencing, but essentially reaping the benefit of comparing our own microbiome and our self-reported traits like ethnic background, the foods that we eat, how often we exercise, whether we get sick regularly or not, and really to try to establish some common links between microbiome composition and such traits across a huge diversity of people. Now, importantly, this microbiome project is now expanded well beyond the US. It involves such places like Canada and even Europe. And any one of you that is interested to participate can follow through this publication to understand how these communities vary and what are some of the important associations that are emerging from the study of this microbial composition. So let's take a look at an example. This is actually a report that one might get from producing that kind of sequencing. So you get a kit and you sample different areas on your body using these swabs, sending them over to a central lab where that sequencing is performed. And based on the analysis of such sequences, you can see how your sample compares to some of the other samples that are already in this database. Now, what we will do in the program is take several samples like these to produce a report exactly like this. So that will give you some hands-on practical work experience dealing with samples that come from healthy and sick people that have contributed their own samples to this database. Now, another important area of research is how does the specific microbiome composition affect different areas of normal, subnormal, and clinical conditions. In some cases, like gut microbiome, we can sample clinical patients and find associations with clinical conditions of the microbiome composition changes. But in many cases, it is very difficult to have a controlled environment where such changes could be studied. And a good example is the link between obesity, which is a phenotypic condition, the microbiome, which is difficult to necessarily link to this phenotypic condition, anxiety, which is a physiological but more a psychological condition, and to really establish a link. And so to address this challenge, there have been a number of animal models that have been developed to really elucidate the direct connection between microbiome changes and such conditions. The way it was done, and we'll take a look at this kind of an example in greater detail, is we'll take this publication that's called Obese Type Gut Microbiota Induce Neurobehavioral Changes in the Absence of Obesity. Now, the way that they performed this experiment is they took early young mice at 3 months old. That's when the microbiome is being developed. They then reduced the bacterial load. So they really reduced the composition, the complexity of the microbiome, did some behavioral testing, and developed a model where you can see how different recipients of microbiome composition from these donors that were at a young age established on a specific diet can lead to actual characterization of changes in microbiome and links to their behavioral changes as well. Now, the way such studies could be done is by employing what's called bioinformatics analysis pipelines. So what do these pipelines actually look like? The way it is done is that at each stage, you get some samples, and these samples represent a community that could be characterized by operational taxonomic unit diversity and composition. So let's take a look at how this is done. To take a look at a practical example, we're going to be using the analysis of 16S amplicon data, which is a study that we just described from that paper, an actual data set from that paper, where you can analyze sequencing data using the data-to pipeline. So to take a look at this practically, please go with me to server.t-bio.info. Okay, this is a platform that we commonly use in our analysis logic. So what is important to understand is not only how this data is produced and what types of studies are out there, what types of data sets are out there, but really to see how does the data become informative? How do we reveal information, and what kind of information is it going to be? So this is the platform that we use for training, but it's also a research tool that you can use in your analysis of metagenomic sequencing data. Now, if any one of you would like to get a quick demo, I'm going to do this to just showcase what it would look like, but you can also request a demo account from Beepsa here on chat, and then you will be able to follow along. So down here, I'm going to just show you this section, the amplicon 16S, 18S section. And we have a demo pipeline here that I will just briefly go through so that we don't have to analyze the data in real time. So this is pre-analyzed data. So this is what's called the data-to pipeline, and it is an important tool for simplifying the diversity by merging high-quality reads into operational taxonomic units that are mapped onto a reference database, and then we can visualize that information using phyloseq. So I'm going to create here a very simple pipeline that will look at hypervariable regions V4 and V9. You can see this is the 16S ribosomal RNA. So there's V4, and here we have V9 right here. These are hypervariable regions that are commonly used to characterize diversity in metagenomic sequences. The data-to pipeline, as I mentioned, is an important tool that allows us to remove all kinds of artifacts that can lead to false positives and false negatives in the way that we characterize the composition. Now, this noise is an important challenge in all metagenomic sequencing. So here what we will do is we will take a few steps to really reduce the noise and make sure that we create cluster samples that are consensus OTUs, remove chimeras, and then use mapping onto a database by assigning taxonomy to these samples. After this is done, we are going to identify relationships between these OTUs by analyzing phylogenetic relationships using APE. So what APE does is it forms a relationship based on multiple sequence alignments of these OTUs. And then we can visualize this data by using a commonly used package in R called phyloseq. So here you can see differences in compositions, and what is useful in phyloseq is that it produces some standard visualization tools, and we'll talk about how that is done in just a second, to help us understand the variability between these different samples. So to illustrate what we will typically get out of it are several PDF files that allow us to visually understand what we have found. So first of all, I am going to look at the scores of read quality. So here you can see the read quality. The read quality is important for us to understand what is the actual level at which the cutoff of the threshold of read quality is done. You can also see the results. So this quality of reads is actually done by the data-to pipeline. And then once we have high quality data, we can start analyzing that using phyloseq. So here we have the phyloseq results. So the phyloseq results allow us to look at composition. What does this composition actually look like? The composition, here you can see it is done by family. So this is a very good level of composition. And you can see how we have diversity between different samples. And so here we have the samples, here we have the abundance, or this is proportional abundance of different families of microbiomes, including some unknown ones that were not characterized. This kind of an abundance can actually characterize diversity based on alpha and beta diversity. So alpha diversity and beta diversity allow us to look at composition within samples and between samples. This is not just simply done based on the abundance of OTUs, but also on the phylogenetic relationships between individual OTUs, representing species richness, or how diverse each sample is and how that diversity changes between groups of samples. So here you can see the Chow-1 and the Shannon metrics for alpha diversity, and you can see how those samples vary in those diversity measures, as well as you can see right here you can see beta diversity, which is done by non-metric dimensionality scaling, which is showing us a real clear difference between the samples that are done on regular diet, that have been grown based on regular diet, and those that have been grown on high fat diet. Really, that illustrates how we can understand the relationship between microbiome composition and the relationship with a phenotype that we can actually characterize or study using a psychological test. Okay, let me just give you a couple more examples, and then I will open this up for questions again. An important part of such studies has allowed us to understand how different types of conditions, including changes in diet and including in the changes in treatment, have led to important discoveries about the microbiome. For example, one such discovery was made on the effect of microbiome changes in the composition and Alzheimer's disease. So in fact, you can see here in 2019, China has approved Alzheimer's disease treatment that targets the microbiome. So these are very important new discoveries that have been done in the context of microbial studies based on an association with human samples, as well as samples that were designed in animal experiments. Another important result of such studies is the effect of cancer therapy. So anti-cancer treatment modalities and co-medications, we know that they affect the integrity of the epithelial barrier. They also affect the actual microbial composition. So you can see here how the different changes in microbiome that affect the immune response, that affect the integrity of different organs and the protective films that these microbiota form, as well as the actual direct impact on the immune cells involved in responses. So as a result, we can identify microbial signatures, as you can see here below, that actually will help determine whether a particular cancer treatment will be effective, and to track the effectiveness of such treatment over time of the course of treatment, potentially changing the course of treatment or seeking to modulate the microbiome composition to prepare, to kind of prep up the patient for effective consumption of treatment for particular diseases. Okay, so these were just a few examples. I know that there are a lot of questions about other types of examples that could be done. So let me focus right now on some of the questions that you've posted on the chat. Beatsa, if you could help me sort through the questions so I could respond to the important ones.
Yes, sure, Ilya. So we had one from Shraddha earlier. So do the microbiome of normal person differ from diseased one?
Yes, so obviously there's a lot of variability between what we call normal people, right? We know that normal people consume different foods. They grow up in different environments, even drinking different water. So all of those things change the way the microbiome develops, as was shown in this example with animal models, where transplanted microbiota actually led to specific types of changes in behavior. Now, those would not necessarily categorize a person as a non-healthy or a clinically sick person, but it definitely changes susceptibility to diseases, changes the response to different pathogens, and as we can see here, changes the response to different treatments. So obviously, there's a lot of diversity to sort through before we define whether there's an association with a clinical condition. Now, some clinical conditions have very clear-cut changes. For example, irritable bowel syndrome or IBS, different other types of sensitivities of the gut. More and more of such changes are weaker links, so they're really just a component in a complex systems biology relationship between a clinical condition and the microbiome. So I think we have to put it into context, if that answers your question. If not, you can maybe clarify. Now, I do see that Modibo here put a question privately to me, so I will just read that out. How can we simulate in silico the repair genes interactions with mutated gene or for DNA reparation, and which server or bioinformatics could do it? So if I understand correctly the question, the repair genes, DNA repair genes, and mutations in DNA is what the question is all about. So maybe a scenario or a project example here is to think of cancer. So we can think of cancer oftentimes caused by somatic mutations. Somatic mutations are repaired by repair genes. And so mutations in repair genes and accumulation of somatic mutations in the DNA eventually causes cancer. Now, how can we simulate the interactions between the repair genes and DNA mutations? So I think that you need to develop a mechanistic model that really defines all of the relationships. But before you define that model should be data-driven. So I think the first part of it is to really take some sequencing data that combines gene expression and mutation data. So you can look at accumulation of mutations or presence or absence of particular somatic mutations and the expression of genes. And really the interaction could be studied by what's called multi-omics integration. The server that I just demonstrated, the server tbioinfo, server.tbioinfo, has a number of sections for both multi-omics integration. So you can see right here, multi-omics integration that seeks to take two different types of omics data and study relationships within this multi-omic data set using different approaches. For example, integration based on partite networks or bipartite networks or clustering and other types of approaches. So that's one way to study the interactions. Once you develop a mechanistic model, there are a number of mathematical modeling tools that we also have, and that typically involves mathematical simulation. So once a mathematical model is developed, you can simulate that model. Now that's a very complex process of actually making that model first and maybe testing the model prediction on data. So if you are interested in that, we do have a section related to that, but I think we would need to understand a lot more what the actual project is about to really help you. Okay, any other questions?
Yes, Celia, we have one from Dr. Manav Jain. Can there be temporal relationship between duration for which microbiome is altered and development of disease? For how long microbiome should remain altered?
So it's a great question. I would assume that question would be more directed to a microbiologist. I myself am not a microbiologist. I more can address the questions about the bioinformatics component here. So I think what would be a good approach to try to answer that question is to find a specific data set. And I'm actually going to get to that point in just a few minutes. So maybe we can take the remainder of the questions and just email them to us. We would be happy to continue this interaction after the webinar to really help you identify project examples, existing data sets, and link them with effective tools that will help you try to find some answers to these questions. And if you are a microbiologist and you're interested in trying to apply metagenomic sequencing data analysis tools to answer such questions, I think there are a wide variety of publicly available data sets. And that's the exciting part, I think, in silico studies and computational biology studies, that we have access to such great microbial data sets that we can analyze. So I will get to that in just a second, in just a few minutes. Sorry. Okay, so we talked about a few examples, and again, thank you for your questions. I think these are excellent questions. I do want to mention that the program that we are going to be launching on November 17th is going to be designed for those of you that have such questions and would like to work with different types of data, including metagenomic sequencing and other types of omics data, to be able to answer such data sets, such questions that you have. So let me talk briefly about this upcoming Omics Logic Metagenomics program that will be starting November 17th. The online program, again, is designed to help students as well as researchers and faculty to try and address such questions with bioinformatics analysis. So the objective of the program is to help you develop an independent project, maybe trying to answer some of the questions that you have already posed in this chat, leveraging the tools and the data that we will discuss and develop practical skills with, so that anyone can actually use them. So the program is designed to really highlight the role of big omics data that is shaping modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration and machine learning are transforming personalization of treatment selection, and innovative algorithms allow for unprecedented capabilities with drug discovery, drug design, as well as repurposing. And so the important part is that these various types of omics data that we describe in great detail, intracellular and intercellular processes associated with common diseases, they have to be analyzed to understand the biology behind host factors and microbiome composition in our bodies and the environments that we live in, to drive infection and cause deterioration of health, to destabilize the immune system, and lead to clinical manifestations of disease. So these multi-omics data sets require different approaches to the processing, integration, visualization, and interpretation. And the objective of the program is to give you a sense of how these different methods and tools could be applied directly to study microbiome or the host responses, to find links and associations with some of the clinical data that we have about patient interactions with their clinicians. So the objective of the program, again, as I mentioned, is to provide a platform for you to think about, find, develop projects that are independent, and get some feedback from participant experts in the panel that will actually help evaluate and help you refine some of the ideas that you might already have. So really, at the end of this program, you will be able to present a project to a panel of experts. Here we will bring in some experts that have experience with metagenomic sequencing data analysis. They will be able to help guide, prepare you, and then evaluate your project proposal. Now, the objective of these Omics Logic programs is to provide you with a clear perspective on big data analytics, machine learning, and artificial intelligence, so that anyone can try and apply a variety of different analysis methods. And to do that, obviously, it's very important to have access to curated data sets. So what we will do in the context of this program is actually look at such public repositories like the Human Microbiome Project, the American Gut Project, We will look at different disease-specific metagenomic data sets, and a lot of the tools that you will gain will allow you to effectively search NCBI, the National Center for Biotechnology Information, and identify data sets that are ready for analysis. Our previous participants have developed a number of research projects together in this program that have led to poster presentations, publications, grant applications for those that are already in research, where you can leverage bioinformatics to define some interesting preliminary results. And those preliminary results can define the way that you are thinking about a particular research question. So to summarize, what we've learned today is an important part in this emerging field of microbiome research that has far-reaching implications for different types of conditions that are both clinical, psychological, environmental, and others. And for you to get started on this journey, we are excited to present this 6-session program. This program is designed to help you get started if you are coming from a life science background or you're coming from a computational background. It's really designed to launch you into this field of metagenomics. So let me describe this program a little bit to explain how you can join and what you can take away from this program. So first of all, the link to the program itself, where you can see in much greater detail all of the different elements that I'm describing now, is metagenomics.omicslogic.com. This program itself, we can take a look here together, what it will cover. You can see the dates for this program, a short overview of the program, and here you will see some of the specific topics that will be covered in each one of the sessions, as well as some of the asynchronous resources and some of the specific project examples that we will go through, as well as some of the tools that we do not have time to showcase in this session because we're already running over time. But you can definitely see a lot more information about this program on this link. Now, briefly going through some of the sessions and what will be covered. These are interactive training sessions. We will talk about the specifics of the program and what type of resources that you will get. These include big data analysis tools on the cloud, include asynchronous resources that you can do on your own time, interactive sessions where we will do assignments together, including coding and running some pipelines, as well as some feedback from experts that will help guide your independent project and refine your ideas as you're trying to look for a particular data set. We will talk in detail about metagenomic sequencing and guide you through an asynchronous course where you can learn about the individual terminology and the different types of concepts that are important to know. We'll talk about mapping on databases and visualizing the sequencing data in a cloud-based environment. Then we'll talk about how to study variability and composition, including some important pipelines that are considered to be the gold standard, like Data2 and Kind2. We'll then talk about some of the examples that we talked about here briefly, the American Gut Project and the Human Microbiome Project, as well as such animal model studies like the microbiome diet and the association with the gut-brain axis. Finally, we will help you develop an independent project from publicly available data sets that you will be able to define and develop a proposal, potentially even getting some preliminary results that you can showcase. Finally, we will talk about some expert feedback for that project. Now, for the program, we will leverage some online tools. And in our next session, when we do the introduction to this program, and that will be a free webinar that all of you are welcome to join, we will actually go through the different types of asynchronous materials and discuss the scheduling and how to get started. So the program will start on November 17th. I hope that this webinar is somewhat informative and I was able to answer at least some of your questions. We do have a blog post on the different types of applications of metagenomics in different types of communities, like the skin, the risk of dementia, autoimmune diseases, some of the ways that you can culture and study bacteria in the lab. So if Bipsa, you could place a link to that blog. So if any one of you is interested in the different types of projects that we will discuss, you will find those discussed on this link. Importantly, I think that what we are trying to establish here as a company is not just the training itself and not just providing you with cloud-based tools that you can use. An important component, I think, of these programs are the community that we are building. And this community includes students, young faculty and researchers, as well as established faculty and professionals that will help you understand the applications of metagenomics and microbiome research in important basic and translational research projects. So we always welcome those of you that are interested in Omics Logic to join our social media groups. And one of them is called the Omics Logic group on LinkedIn. You can see here the link to the LinkedIn group. In fact, I'm going to just copy these and paste them here in the chat, as well as a Facebook group that you will see includes thousands of participants from around the world that will join some of the programs that we have. And there are many different programs that we have throughout the year, not just focused on metagenomics, but also focused on other omics fields, as well as data science skills and specific research areas like infectious diseases or precision medicine. Thank you, Bipsa, for the blog. Now, I want to leave you with a couple of links that I've used throughout this presentation. An important link that you will find here highlighted is a full timeline of milestones in human microbiota research published by Nature, but you will also find some of the other links that I have highlighted in this presentation. And these include a couple of articles, review articles on the current and future topics in metagenomics and microbiome research, as well as some of the tools that I briefly mentioned in this program and in this webinar. Okay, so as a conclusion, I'm happy to address any of the questions that I might have missed. So please let me know if you have any additional questions about the metagenomic sequencing, microbiome research in human health, or the program that is upcoming, Metagenomics, omicslogic.com.
Yes, Ilya, we have 3 questions. So one is from Shraddha. So her question is, I think a person with breast cancer or any other cancer or disease have some sets of microbiome niche compared to normal ones. Can it be used as biomarker or predictive markers by sequencing and finding out novel ones?
Okay, so I think, as I mentioned, there are, and maybe I'll use this opportunity to maybe demonstrate this. So if we briefly go to, if we briefly go to NCBI, and I hope that this is going to be useful for the other participants here as well. Let me put this link in chat. I'm sure that most of you are familiar with it. So here you can see on NCBI, this is the National Center for Biotechnology Information. It contains a lot of different repositories that you can access for free. So in here, you can do a quick search, right? And so your question is about breast cancer and microbiome. So I'm going to just search this and see all the different types of databases that contain information on the relationship between breast cancer and microbiome. So here you can see literature, so different publications, genes, proteins, clinical, and genomes. So here what I would do is I would go to this bioproject section. The bioproject section is actually a definition of different types of projects that have some data associated with it, so you can actually take a look at the data. So here you can see, for example, different types of brief descriptions. Okay, so here is a great example of what your question is about: microbiota of breast cancer tissue. Here you have human metagenome comparative analysis of breast tumor microbiome in Black non-Hispanic and white non-Hispanic women. Gut microbiota profiling in patients with HER2 negative metastatic breast cancer receiving metronomic chemotherapy. The human tumor microbiome of aseptically collected human breast tissue in benign and malignant disease. So you can see here that there is a very big diversity of the types of data sets that you can get. For example, you have treatment versus non-treatment. You have different patients, maybe at different types or subtypes of breast cancer, maybe in different kind of compounding factors like ethnic diversity and background. So you really have a variety of data sets that you can start looking at to ask these specific questions. Most of these will actually have a publication associated with it, or will describe how the data was collected and who was collected by and what it was collected for. So really you can start looking at some of these examples. Some of them are very recent, June 2020, so very up to date. Some of them might be a little bit older. So here you can see a data set, okay, also 19th of August. So maybe these don't even have a publication yet because they're so recent. Let's take a look over here. Weizmann Institute of Sciences in Israel. So here you also have April 2020. So this is very cutting edge research. I encourage you to take a look at some of these examples. And in the program, we'll actually do this kind of analysis together. Any other questions?
Yes, Ilya. So we have another question from Gaurav Patekar. He wants to know if the environment plays an important role more than the genetics for development of the microbiome, because the microbiome study is not done much in respect to genetics.
Yeah, I think that's an excellent question. So as we see, the microbiome develops in a much more variable way than genetics. And a great, excellent study for this is the study that is called, they are called twin microbiome. Okay, so there is a twin microbiome study that recently, not so long ago, has shown how different factors actually influence the development of microbiome in genetic twins and twins that are genetically identical, right? And so that I think directly answers your question, how the effect of environment, the effect of different preferences for food, maybe how they grew up in different environments at different stages of their life really has an effect on the gut microbiome. And I think that is an excellent place to start answering your question. So probably I'll just paste the link to this publication right here. There's a whole project here. They have clinical data, microbiome data, genetic data. So there's really a lot of data here to go through if you're interested in that topic.
Thank you, Ilya. And we have one last question. So this one was from someone whose name is still iPhone. So the question is, my research scope is on lung-brain microbiome axis using RSV virus on animal model. What bioinformatics approaches are the most suitable?
So of course, I would love to know a little bit more about your research project, just kind of taking that information from that brief description. As I understand, you have a virus. So RSV is a respiratory virus, if I understand correctly. And then some of these viruses actually replicate in the central nervous system. And I might imagine that that is the relationship between the lung and the central nervous system or the brain that you are referring to. When we talk about the gut-brain axis, it's a little bit different. It's the effect of how the gut microbiota can cause changes in behavior or in psychology. So when you talk about a virus, a pathogen that directly affects neuronal cells or has a path of entry that goes through multiple organs, for example, intranasal cavity or lungs and then go somehow to replicate in the neuron cells, that's very different than the effect of a specific condition, and perhaps through kind of this quorum sensing passed on to other organs in the body. So if you are looking at virology, you're looking at virus sequences, viral replication, we do have a completely different program called Bioinformatics for Infectious Diseases. So Bioinformatics for Infectious Diseases. Okay, so this is a program that's actually starting on November 9th that I would recommend for you to explore. Here we do talk about the specific tools for viral sequence analysis, so viral genomics, phylogeny of viruses, evolutionary studies of viruses. We'll talk about things like tissue tropism and how specific mutations in viral genomes could be associated with the way that they replicate in different types of cells. So I think that would be a more suitable direction if you're interested in that particular project that if I understood it correctly. Okay, so again, thank you everyone for joining today's session. I am going to stop the sharing. If you do have any last questions, please do let me know. I would be happy to continue this conversation. We will be starting this program again on November 17th. And we will have our first couple of sessions are going to be again open and free for anyone. So you will be able to join just to evaluate and be able to continue in asking these excellent questions that we've had in this session. So again,